Welcome to the deep dive. Today we're jumping into something pretty interesting, maybe a bit alarming actually, the Oropouche virus, or ORV.
Right.
Picture this, a mosquito-borne illness, usually confined to the Amazon region, but now it's spreading fast.
Yeah, we're seeing cases pop up way outside its usual zone. Colombia, Peru, Bolivia, even Cuba.
And the numbers, they're forecasting a big increase in infections, like through next year.
It's significant.
But here's the real kicker. This thing causes serious problems, fever, neurological damage, pregnancy complications, yet it's still considered, well, largely neglected.
That's exactly right. And that's why for this deep dive, we're digging into a really important new scientific paper. It's called MyD88 signaling in B cells and antibody responses during Oropouche virus-induced neurological disease in mice.
Sounds technical, but
Yeah.
basically it looks at how our immune system fights back, right?
Precisely. It's by Toledo Teixeira and colleagues, just published in eBiomedicine.
So, our mission today for you listening is to sort of cut through that complexity. We want to show you how our bodies are actually defending against RRAV.
Yeah, and uncover some pretty surprising roles for certain immune cells and pathways, things that might actually point towards new treatments or vaccines down the line.
Exactly. Should
Yeah.
be fascinating. Okay, so let's unpack this a bit more. We said ORV is spreading, but what is it fundamentally and why the buzz now?
Okay, so ORV is what's called an orthobunyavirus. It's an RNA virus, trisegmented negative single-stranded RNA to be specific, and it's carried by insects, so a vector-borne virus.
And it's not exactly new, is it?
Not at all. It's been endemic, sort of consistently present in Latin America and the Caribbean for, well, decades. We're talking probably more than 500,000 cases since the 1960s, maybe more.
So why the sudden spotlight?
It's this recent spread, seeing it establish itself outside the Amazon, like in the places you mentioned earlier that's unexpected, it suggests the virus might be adapting, or maybe environmental factors are helping it move. That's why it's getting global attention now.
Okay, so if someone gets infected, what does that look like? What are the symptoms?
Well, for most people, it starts as an acute febrile illness, basically a sudden high fever.
Right, like the flu.
Similar, yeah. Often comes with headache, muscle pain, joint pain, myalgia, arthralgia, maybe light sensitivity, sometimes increased urination, even a skin rash.
How long does that usually last?
Typically about a week. But, and this is important, for a lot of people, the symptoms can linger, or they might feel better and then relapse.
And it can get worse than that, you mentioned.
Yes, definitely. ORAV can be lethal in some cases.
Yeah.
And tragically, it's been linked with problems in pregnancy, fetal abnormalities, miscarriages.
Wow.
And then there's the neurological aspect. It can actually invade the central nervous system, the spinal cord.
Causing neurological disease in adults, too.
Yes, in adults and in unborn children. That neuroinvasion is a really serious feature.
That sounds incredibly serious. How does it actually jump from, say, animals to people?
So its main reservoir is in what we call a sylvatic cycle, a cycle in wild animals. Think sloths, maybe some non-human primates, birds, rodents. They act as amplifier hosts.
Meaning the virus multiplies well in them.
Exactly. Humans, on the other hand, we're generally considered dead-end hosts.
So we get sick, but we don't really pass it on effectively.
Typically not in a way that sustains major human-to-human outbreaks. The main way we get it is through the bite of an insect vector. The primary one in urban settings is this tiny biting midge, Culicoides periensis.
Well, biting flies, basically.
Yeah. But other mosquitoes, like Aedes serratus and Culex quinquefasciatus, have also been implicated.
And you mentioned the brain invasion. Do we know how that happens?
Well, that's been a big question mark. The precise mechanisms of how it gets into the nervous system and how it causes damage there, that's remained pretty obscure.
Which is where this new study comes in, I guess.
Exactly. It tackles that very problem, understanding the host immune factors involved.
Okay, this is where, for me, the deep dive gets really interesting, the immune response. Before this study, what was the general thinking? What did we assume our bodies did to fight OROV?
Well, you know, for a lot of these arboviruses, viruses carried by insects like mosquitoes or midges, the first line of defense we often look at is the type I interferon system.
Right, the body's sort of early warning system.
Exactly. Interferons, IFN for short. And previous research did show that mice that couldn't respond properly to type I interferons, specifically mice lacking the IFN alpha-beta receptor, they were very susceptible to Orvo. They got sick and died.
So interferons are important.
They are. But there was this little wrinkle in earlier findings. They looked at mice lacking different components of the interferon signaling pathway IR3 IRF5 and 7, and mice lacking IRF5 seemed particularly vulnerable to neurological disease.
More so than others.
Yeah, more so the mice lacking IRF3 and 7 together. And this increased susceptibility in the IRF5 deficient mice was linked to having lower levels of neutralizing antibodies against the virus.
Ah, antibodies. So that was the clue.
That was the hint, yeah. It suggested that maybe antibody responses, which are driven by B cells, might actually be playing a key protective role. It wasn't just about interferons.
Okay, so the researchers behind this new paper decided to zero in on that. Was it B cells, the antibody makers, or T cells, another key immune player, or maybe both, doing the real protective work against ORV? How'd they figure that out?
They set up a really neat comparison using different mouse models. They had their standard healthy wild-type mice. Then they had mice called RAG1 knockout.
Mm-hmm.
These guys lack all mature B and T cells.
So no adaptive immune system, basically.
Right. Then, critically, they had two more groups.
Right.
One called M and T mice, which specifically lack mature B cells but have T cells.
Okay.
And another group called TCR mice, which lack most mature T cells but do have B cells.
Clever. So you can isolate the role of each cell type.
Exactly. They infected all these groups with ORV and then watched closely looking at weight loss, clinical signs of illness, survival rates, and how much virus was in their bodies.
And what jumped out at them? What was the first kind of aha moment?
Well, the RAG1 knockout mice, the ones lacking both B and T cells, they did very poorly, as you might expect.
Got really sick?
Yeah, severe weight loss, clear signs of neurological problems, lethargy, paralysis, difficulty moving, ataxia. And when they looked, they found really high levels of OROV in their organs, especially the brain and spinal cord.
Okay, so the adaptive immune system is definitely needed. But the B cell versus T cell question?
That's where it got really stark. The wild-type mice, the normal ones, were resistant. And importantly, the TCR mice, the ones without T cells, but with B cells, they were also resistant. They handled the infection just fine.
Wow.
But the MNT mice, the ones lacking B cells, they all succumbed to the infection. They developed neurological signs and died, usually between 1 and 3 weeks after getting the virus.
So it wasn't the T cells that were essential. It was the B cells.
Precisely.
That is a pretty significant finding, isn't it? Because often T cells get a lot of the credit for fighting viruses.
It is. It really shifts the focus here and says B cells are absolutely critical for controlling ORV infection and preventing that severe neurological disease. T cells, surprisingly, seem dispensable in this model.
Okay, so if B cells are the unexpected heroes here, the next question is how? What's going on inside those B cells that makes them so protective? You mentioned that earlier clue about IRF5 and antibodies.
Right. And remember, IRF5 is often activated downstream of toll-like receptors, or TLRs, via an important adapter protein called MyD88. TLRs and MyD88 are part of the innate immune system's sensing machinery.
So they sense the virus.
They can sense viral components, yes.
Yeah.
And this connection, IRF5 being important, IRF5 being linked to MyD88 signaling, led the researchers to hypothesize.
Yeah.
Could MyD88 signaling specifically within B cells be the crucial switch? Is that the key mechanism?
Okay, makes sense. So how did they test that specific connection? Did they find MyD88 was the key?
They did something quite sophisticated. They generated mice where the MyD88 gene could be specifically deleted only in B cells. They used a technique involving CD19 Cre, which targets B cells, and floxed MyD88 alleles.
Right, so MyD88 works fine everywhere else, but not in B cells.
Exactly. And the results were, again, very clear. These mice, the CD19 Cre plus MyD88 B4L mice, when infected with ORV, they developed weight loss, neurological signs, and they died. Just like the mice completely lacking B cells.
And they had virus in the brain.
Yep. High levels of virus in their blood, organs, and central nervous system. They could even see ORV antigens, parts of the virus, within the brain tissue.
And the control mice.
The control mice, which either had normal MyD88 everywhere or lacked the Cre part needed for deletion, they were completely fine. No disease signs, no evidence of ongoing viral replication.
So that really pinpoints MyD88 signaling in B cells as essential for protection.
Absolutely essential.
And just to be thorough, they double-checked that other pathway you mentioned earlier, the type I interferon pathway within B cells.
They did. They created mice where the type I interferon receptor, IFNAR, was knocked out specifically in B cells. Those mice were completely resistant to ORV.
Okay, so it's definitely not type I IFN signaling in B cells that's required. It really is MyD88.
It strongly points to MyD88 as the critical pathway within B cells for controlling this specific virus.
Interesting. And did this lack of MyD88 in B cells affect the overall inflammation, like the early warning signals?
That's another good point. They checked, and actually, no. Even the mice lacking B cells altogether, or the ones lacking MyD88 just in B cells, they still produced a normal range of inflammatory signals, chemokines and cytokines like IFN alpha, IFN beta, IL-1 beta, IL-10, IL-12, CXCL1 after infection.
So the general alarm still goes off.
Right. It suggests MyD88's crucial role in B cells isn't about triggering that initial broad inflammation, but something more specific to the B cell's function itself, likely related to making those protective antibodies we talked about.
Okay, that makes perfect sense. So B cells are vital, and the MyD88 signal within them is the key switch. Now let's connect that back to the antibodies. How exactly do these MyD80 activated B cells protect? Is it all about producing the right kind of antibodies?
It certainly looks that way. Antibodies seem to be the main weapon here. The study showed that normal, wild-type mice started making OROV-specific antibodies pretty quickly, like within 6 days after infection.
And good antibodies, effective ones.
Very effective. By day 14, they had really high titers, high levels of neutralizing antibodies, the kind that can directly block the virus from infecting cells. The measurement they used, FRNT50, was like above 1 to 65,000, which is really potent.
Wow. And what about antibodies we might just have lying around normally? Did they check if those help?
They did. They looked at natural antibodies from mice that had never seen OROV. And nope, those weren't sufficient to protect. You really need that specific adaptive antibody response triggered by the infection itself.
Okay, so the body has to actively make these specific antibodies after getting infected. What about the timing? You mentioned earlier studies hinted at timing being important.
Absolutely critical. They did a serum transfer experiment, which really highlighted this. They took serum, the liquid part of blood, containing antibodies from infected wild-type mice at different time points.
And gave it to susceptible mice.
Right, they gave it to the RAG1 knockout mice, the ones with no B or T cells. If they transferred serum collected early, specifically within 3 days of the original infection, it protected the RAG1 mice from developing neurological disease.
Okay, early intervention works.
But if they transferred serum collected later, say at day 6 post-infection, it didn't protect anymore.
Really? Only a 3-day window?
It reveals this surprisingly narrow time window where antibody therapy, or maybe the body's own early antibody response, is most effective at preventing the severe neurological consequences.
That's a crucial detail. And did they figure out which type of antibody was doing the work in that early window?
Yes. They suspected IgM, which is typically the first antibody type produced during a new infection.
How'd they confirm that?
They treated the protective serum with a chemical, OMEA, which specifically breaks down IgM antibodies. And when they did that, the serum lost its ability to protect the RAG1 mice.
So it really was the IgM. Early IgM is vital.
It seems essential for that early control, yes.
Okay, putting it all together, early IgM is critical. There's a narrow window, and MyD88 signaling in B cells is needed for protection. So how does MyD88 link to these antibodies? Does it just tell the B cells to make more antibodies, or does it affect the quality too?
That's the key question, and the study suggests it's very much about the quality and effectiveness. The mice that lacked MyD88 in their B cells, the CD19 Cre plus MyD88 they did produce some antibodies against ORV.
Oh, so they weren't completely unable to make them.
No, but the antibodies they made were, well, suboptimal. They had lower titers overall, and crucially, the antibodies had reduced neutralizing potency compared to the antibodies from mice with normal MyD88 signaling.
Can you be more specific? Like, was the early IgM affected?
Yes. On day 6 post-infection, these MyD88 deficient B cell mice had lower levels of OROV-specific IgM, and that IgM also showed reduced avidity, basically. It didn't bind as strongly to the virus.
And later antibodies.
Same issue. By day 14, their levels of ORO-specific IgG, another important antibody type, were also lower. And perhaps the most direct proof, when they took serum from these mice and transferred it to the susceptible RAG1 mice.
It didn't protect them.
Exactly. It failed to confer protection. So MyD88 signaling in B cells isn't just an on-off switch. It's critical for generating a high-quality, potent, and timely antibody response, especially that crucial early IgM.
Right. So MyD88 is like a quality control manager for the antibody production line. Fascinating. So if MyD88 affects the antibodies, does it also affect the B cells themselves? Like, are certain types of B cells more reliant on this signal?
That's exactly where they look next. And yes, they found that MyD88 signaling was crucial for the development and maintenance of specific B cell populations, particularly during the ORO infection.
Which populations?
In the mice lacking MyD88 in their B cells, they saw significantly fewer marginal zone B cells and fewer plasmablasts after infection compared to normal mice.
Okay, remind us what those are again. Marginal zone B cells and plasmablasts.
Sure, marginal zone B, MZB cells, are a type of B cell primarily found in the spleen, kind of poised for rapid response to blood-borne antigens. Think of them as maybe the B-cell first responders. Plasmablasts are immature plasma cells, basically differentiating B cells that are gearing up to become high-rate antibody-secreting factories.
Got it. So fewer first responders and fewer antibody factories in the MyD88 deficient mice.
Exactly. In contrast, in the normal wild-type mice infected with ORV, both the MZB cells and the plasmablasts actually expanded in number. The immune system was ramping up production.
And did the analysis point to one of those being particularly important?
Yes, their statistical analysis, something called factor analysis of mixed data, actually highlighted the marginal zone B cells as the primary cellular difference between the protected wild-type mice and the susceptible MyD88 deficient B cell mice.
Okay, this is where it gets really cool. Did they actually prove that it was those specific cells, the MZB cells, that were making the difference?
They did, with a really elegant experiment and adoptive transfer focusing specifically on MZB cells.
What did they do?
They isolated marginal zone B cells from the spleens of either naive wild-type mice with functional MyD88 or from naive CD19 Cre plus MyD88 aval mice lacking MyD88 EB cells. Then they transferred these isolated MZB cells into susceptible mice,
Into which susceptible mice?
primarily into the CD19 Cre plus MyD88 ME cells, the ones that normally die because their own B cells lack MyD88. They also tried transferring them into the RAG1 mice lacking all lymphocytes.
And the result. This sounds like the clincher.
It really was. The CD19 Cre plus MyD88 mice that received healthy, wild-type MVB cells were fully protected from OROV disease.
Wow. Just giving them the right kind of B-cell precursor fixed the problem.
Exactly. And even more remarkably, some of the RAG1 mice, the ones with no immune system, also survived the OROV challenge if they received these healthy, wild-type MZB cells.
That's incredible. And did they check antibodies on those rescued mice?
They did. The protected CD19 Cre plus MyD88FL mice that got the WTMZB cells had significantly higher levels of neutralizing antibodies compared to those that received MyD88 deficient MZB cells.
And what happened if they transferred the MZD cells that lacked MyD88?
As expected, transferring those MyD88 deficient MZB cells offered no protection at all. The mice still succumbed to the infection.
Okay, so that just nails it down. MyD88 signaling is crucial in marginal zone B cells, enabling them to respond effectively, likely differentiate into plasmablasts, and produce those life-saving, high-quality, early neutralizing antibodies needed to stop OROV neurological disease. It's a whole chain reaction.
Precisely. It paints a really clear picture. These early innate-like functions of B cells, especially MZB cells, are working together with the later adaptive antibody response. And MyD88 is the key regulator orchestrating this B-cell differentiation and the production of those critical neutralizing antibodies right at the start of the infection. That's what allows the virus to be cleared before it causes severe disease.
This is fantastic science. But stepping back, what does this all mean in the real world? What are the implications of this deep dive?
Well, I think the implications are potentially quite significant. Understanding this specific protective mechanism, MyD88 MZB cells, early IgM, it gives us very specific targets.
Targets for what? Treatments? Vaccines?
Both,
Yeah.
potentially. This knowledge could directly inform the development of new antibody-based therapies, maybe focusing on delivering potent neutralizing IgM early on, or it could guide vaccine design, aiming to specifically elicit that type of strong, early MZB cell-driven response.
Which we currently don't have for OROV, right?
Correct. There are no specific antivirals or vaccines for OROV right now.
Hmm.
So given that it's an emerging pathogen causing increasing concern globally, findings like these that offer concrete strategies are really valuable.
Now, the researchers themselves, they were quite clear about the limitations of their study, which is always important in science, right? It tells us what we still need to figure out.
Absolutely. Science is iterative. They pointed out a few key areas for future work.
Like what?
Well, for instance, they didn't fully dissect the exact molecular signals upstream of MyD88 that trigger this response in B cells, or the precise downstream pathways MyD88 uses to boost antibody secretion.
Okay, the finer details of the signaling.
Right. They also didn't look at the clonal repertoire, meaning the specific variety of B cells responding, or the detailed gene expression profiles within the B cells that lacked MyD88.
And this was all in mice, wasn't it?
Yes, a crucial point. These findings need to be validated and explored in the context of human ORV infection, and potentially with different strains of the virus as well.
Makes sense. Anything else?
They also mentioned they didn't do a comprehensive long-term evaluation of memory B cells.
Ah, the cells responsible for long-lasting immunity.
Exactly. Understanding how memory responses are generated after OROV infection and whether MyD88 plays a role there too, that's really important for developing vaccines that provide durable long-term protection against future infections.
So definitely more work to be done, but this study provides a huge leap forward and a really solid foundation.
Without a doubt.
So this deep dive has really peeled back the layers on our immune system's fight against Oropouche virus. It's shown us how intricate it is, but also how specific research can pinpoint these critical vulnerabilities or defense strategies.
Yeah, the MyD88 pathway in B cells, the role of marginal zone B cells, that early punch of IgM antibodies, these are now clearly marked as key players.
It really highlights the importance of that early response. So here's something to think about. How might this deeper understanding of very specific immune pathways like MyD88 signaling in B cells change how we approach other emerging viruses? Could focusing on boosting these precise early B cell responses be a key strategy for getting ahead of future viral threats before they can really take